All right, so welcome back. Last thing we were talking about were these lymphocytic cells or lymphocytes, specifically B cells that can become plasma cells, the cytotoxic T cells, the helper T cells, and the natural killer cells. So let's start to get into the specifics of how these cells function within the acquired immune portion of the immune system. Okay? So first, a couple of things. The B cell and the T cell. Let's start here with a B cell. A B cell has located within it these interesting looking receptors that I often jokingly say are one of the few times biologists try to get some sense into what they do, but these particular receptors are associated with B cells, so they're called Bcell receptors. So there is a unique receptor type to a B cell. There are also receptor types that are specific to T cells. Different shaped receptor. So this is a T cell. There is a T cell receptor. Okay? Now, these receptors are capable of binding things, specifically proteins. Proteins can be held within the B cell receptor, and I should say peptides are held within the B cell receptor, not specifically in the receptor like I just drew it. That's why I erased it really quickly. But they bind here, interestingly enough. And the things that are bound are little tiny pieces of proteins. So they're little tiny peptides, maybe not big enough to call a polypeptide, but little tiny pieces of peptides that have come from larger proteins. B cells are also capable of binding these little bits of peptides, okay? And they bind them through the T cell receptor. What's interesting is if you look at T cell receptors are very good at binding peptides that are foreign, meaning they're not peptides that we make, they're somebody else's peptides. So these could be peptides from a surface receptor on a bacteria, or from a protein on the surface of a virus, or a parasitic amoeba, or other eukaryote, or a fungus, right? So the T cells actually go through a process in us in utero where the T cells are checked to make sure that they don't recognize any proteins that we make, okay? Because the idea is they should only be able to recognize the foreign stuff, because then it's going to trigger this acquired immune response. If we bound our own proteins, then we would trigger an immune response against our own proteins. As an aside, that's the autoimmune diseases work exactly this way, where the body begins to mount an immune response against self-peptides. But the system, the way it's supposed to work, is these T cell receptors can only bind foreign peptides. So that's the first piece of information. The second piece of information here is that we make lots of different types of B cells. The types of B cells differ from each other based on the shape of this receptor. So we have armies of B cells that make different types of B cell receptors. They're all the same on this end here, but they differ from each other based on the shape of the part of the receptor that actually binds the peptide. Is that okay? So think about a group of B cells, each recognizing different shaped peptides. And then we have a group of T cells out there that only bind foreign peptides. So far, so good? Okay. All right, so let's go ahead and talk a little bit about how to get these two cell types to interact with each other. Okay. And then to talk about that, we have to talk about another system that occurs within cells that has to do with two different types of MHC class systems. So MHC is short for major histocompatibility complex 1 and major histocompatibility complex 2. The MHCs are proteins on the surface of cells. So here, let's say this is an MHC class 1 protein. All cells have major histocompatibility complex class 1 proteins. All cells have this. Now, the way this works within cells is using the endomembrane system. We're going to build the major histocompatibility complexes through the endomembrane system by using rough endoplasmic reticulum. The protein is now inside the endoplasmic reticulum, then we butt it off into a Golgi, and so on, all the way out to the cell membrane itself. But if you notice in here, the major histocompatibility complex 1 pathway is here. So in the rough endoplasmic reticulum are these MHC class 1 proteins. That's where they're built. Right? The ribosome builds the protein on the inside, right, or builds it and pushes it toward the inside of the membrane. Now, we also have proteins that are manufactured in the cytosol. These proteins that are manufactured there, a small number of these proteins is then processed through this, what's called the endogenous pathway. These proteins are picked up by what's called a proteasome, and they're pushed through a protein called the TAP protein, and now little bits of this protein are now inside the ER, and they bind to these MHC class 1 protein receptors. So this is chopped up, pushed through, binds, and now we have a vesicle leaving the endoplasmic reticulum with an MHC class 1 plus a little peptide on it. And this peptide is from a protein that was made inside the cell. Okay? Then this peptide here, the vesicle fuses with the Golgi and then leaves the Golgi, and eventually this protein ends up on the surface of the cell with the associated peptide attached to it. Okay? Now, the class 1 endogenous pathway is an important pathway because there are many pathogens that one of their tricks is to hide from the immune system by getting inside cells. Okay? Examples of this would be all the viruses. Okay? Viruses parasitize cells. They inject nucleic acid into the cell, and then the cell uses that nucleic acid to build viral proteins. So all viruses hide in cells. There are bacteria that do that as well. Tuberculosis is the one that comes to mind. That's caused by a mycobacterium, and that mycobacterium hides within cells. So the MHC class 1 endogenous pathway is all about trying to figure out if a cell has been co-opted by a virus or by tuberculosis. So not tuberculosis, but mycobacterium. So because this nucleic acid is going to code for particular viral proteins, these viral proteins, some of them will end up being processed by the endogenous pathway, and then on the surface of this cell will be this little viral protein, little piece of a viral protein. Now, the cytotoxic T cells, they can, using their receptors, right, using these T cell receptors, it can target that little peptide. And if this peptide sticks to that, then we know that's a foreign protein, because the self proteins, the ones that the cell makes that we think of as human proteins, should not be recognized by T cell receptors. We go through a process of checking all the T cell receptors in utero. We should have come out of that with no T cell receptors that bind to self protein. Therefore, this cytotoxic T cell knows now that this cell has been taken over or co-opted by a pathogen. The cytotoxic T cell will then target this cell for destruction. Okay? I have that image here. The killing action of cytotoxic T cells. Here is the class 1 MHC in green. The little gold triangle is one of these peptides made by a virus or a bacteria that's inside the cell. The T cell recognized that particular peptide and now mounts an immune response to it. The immune response is to manufacture these little proteins that when they group together, they make pores through membranes. So here are these molecules being secreted. They make holes through the membrane, and now the cell can no longer determine what goes in and out. Everything goes in and out based on diffusion, osmosis, and the cell then dies because of this inability to regulate what goes across the membrane. So the endogenous pathway is used by the cytotoxic T cells to determine if there are any pathogens hiding inside cells. And again, this is a trick of a lot of viruses, and some bacteria do this as well. Okay? All right, so the MHC class 1, every cell in your body puts the peptides that it makes in the MHC class 1 receptors on the surface of the cell, and then these cytotoxic T cells wander around checking these. If they bind to it, right, you should draw it like this. If the cytotoxic T cell binds to the cell, that means that that's a foreign protein, and now we've got targeting for destruction, right, as what's in goes out and what out comes in. Okay? All right, the MHC class 2 pathway. Okay? The other pathway here is a little bit different. Only certain cells have the MHC class 2, and these cell types are the ones that do phagocytosis. So, they would also have a class 1 pathway, but associated with the class 1 pathway is what's called the class 2 pathway. So they manufacture the class 2 molecules, but in the system, the way it's set up is that the receptor is blocked so that it can't pick up any self proteins. Right? You clog the receptor so you don't pick up any self ones. Then you send it in a vesicle to the Golgi. The Golgi sends it off to the lysosome. The lysosome has fused with a food vacuole that contains a bacteria. So let's do some phagocytosis here. All right, here's my bacteria. I bring it into the cell, right? And now my bacteria is in a compartment here. This is the vacuole. Then I fuse that with a lysosome, the lysosome dissolves the bacteria through hydrolysis, breaks it down, and then I'm going to fuse this whole thing that I've built, I'm going to fuse that with my little receptor. Whatever little pieces of peptide get made through hydrolysis can be picked up by the MHC Class 2. So here, my MHC Class 2 is picking up peptides that came from some bacteria or virus that got chewed up by a macrophage or a neutrophil, right, or a dendritic cell. Okay? Now I take this, push it out to the surface, and now I have that little bit of foreign protein in the MHC Class 2 that's being exposed to the outside of the cell. Okay? Now, one of the cell types that loves to cruise these phagocytic cells and look at these particular peptides are these little T helper cells. The T helper cells wander around the body looking for these little peptides. One of their favorite places to find them is on these MHC Class 2 molecules that have the peptides from the foreign proteins that were degraded in the lysosome of the phagocytic cell. The phagocytic cell takes in a bacteria, chops it up, takes little pieces of the peptides, puts them on the MHC Class 2, and cycles them out to the membrane, and the THC cell checks them. Okay? Now, again, if the T helper cell recognizes that thing, right? If it recognizes this, it will start a different process. Okay? Now, any peptide can be checked by the T cells, either a cytotoxic or a T helper cell. But the ones that are of interest to us are the ones that trigger an immune response, either causing the cytotoxic T cells to get all jazzed up, or getting the T helper cells all jazzed up. Any little peptide that has the ability to elicit this immune response